So as I said, we're gonna now start the module 2 component of the 2023 paper, right? So it is the biosystems maintenance question. And here now, figure 5 shows the oxygen dissociation curve for adult hemoglobin at low carbon dioxide concentrations in the lung capillaries of a mammal, right? So the shape of the curve in figure 5 is described as a sigmoidal curve. I'm not sure why they provided that information. It's something that we should have known from the syllabus, but okay, giving us that information. All right. So we know on the y-axis we're gonna have the saturation of hemoglobin, and the oxygen saturation of hemoglobin rather SAO, and we're gonna have the partial pressure of oxygen on the x-axis. In this time, it's in kilopascals. So the first thing we're looking at now, we're asked to explain how the loading of hemoglobin with oxygen gives rise to the sigmoidal curve. So how does the loading of hemoglobin with oxygen give rise to a sigmoidal curve? Anyone? Go ahead.
Sir, it's because the oxygen atoms are going to continuously bind to the iron atoms in the hemoglobin, and it's going to become easier for the oxygen atoms to bind.
So we're looking at well, yeah, so it's most likely that, right? So as we bind oxygen to hemoglobin, it becomes easier for another successive oxygen to bind, right? So it changes its tertiary, well, its quaternary structure. It changes conformation and allows another oxygen molecule or oxygen atom in this case to be easily binded. So if we have no oxygen on the hemoglobin molecule, there's gonna be a general difficulty of binding, right? If we add one oxygen to the hemoglobin molecule, then that will make it much easier for the second to add, then the third to add, then the fourth to add, right? So we're thinking of it like that. So we're looking at the saturation, right? Once a small amount of hemoglobin is saturated, because of that relationship there, because it becomes easier to add successive oxygen, right? Once we add sufficient, right? Once we have sufficient hydrogen, not hydrogen, hemoglobin saturation, it's going to actually skyrocket until plateauing out, right? So that huge jump from around 1 to around 6 there about, that is due to that. So it contributes that sigmoidal curve there. So usually it's a bit difficult at first as partial pressure of oxygen increases. After around 1 to 2, we're gonna see it skyrocketing because of that nature of the hemoglobin conformational change. And then at 8, it's going to actually head towards a plateau. So that's how it creates that sigmoidal curve shape, right? 2, the steepest part of the curve in figure 5 is between partial pressures of 2.5 and 5 kilopascals. A small decrease in the partial pressure of oxygen results in a steep drop in the saturation of hemoglobin. Explain the importance of the curve between the partial pressures 2.5 and 5 kilopascals to mammals. So what's the importance of that part between the curve? We're looking at this area. So 2.5 to generally 5, right? Looking at this area, what is the importance of this area, right, in mammals? Let's read the question once more. All right, let's have a look at the question once more. So it says to explain the importance of the curve between these areas in mammals, right? Are we looking at that if there's a small decrease in oxygen partial pressure, there's a steep drop, right? So between these areas, right? Let's get the markings back real quick. Hopefully we understand what the question is talking about, right? So if we have a drop from 5 to 4, right, we're going to have an exponential drop like this, right? So the partial pressure of oxygen is especially sensitive with hemoglobin saturation at this point, this part of the curve, right? All we're asked to do is discuss the importance in mammals specifically. Just the importance of that curve in mammals. Okay, it's taking a bit of a while. I'm not sure. Do you guys have any idea of what we're supposed to answer here? We're looking at the importance of this region, right, to the entire to mammals specifically. So looking at this region here, what's the importance of this region to mammals? Go ahead.
Sir, I'm not entirely sure, but is this the part where the oxygen is really being loaded in?
Okay, so we could describe it. Yes, it's generally the part. Oxygen is being loaded in all over, right? Within the graph. But we're looking at how the relationship between partial pressure and the saturation of hemoglobin is in this section. It's a steeper curve. What do these things mean? How do we interpret the graphs? Go ahead.
A small increase in the partial pressure leads to a larger increase in percentage saturation of hemoglobin of oxygen.
Saturation, the oxygen saturation of hemoglobin. Okay. Okay. Any other ideas? So that's the interpretation of curve. Now, why is this important? Let me check the chat to see if there are any descriptions in the chat that I'm not cognizant of. Nope, nothing in the
Excuse
chat.
me, sir.
Go ahead.
Sir, could it be that a larger pressure is required because that's where oxygen is really being loaded in and it has to be able to diffuse properly. So by increasing the pressure, um, it makes it easier for the oxygen to diffuse.
Okay, hold on. What does okay? Let's stop there, right? What does partial pressure of oxygen speak to?
Sir, the pressure at which oxygen is transported at.
Okay, not necessarily no. So I think that's the thing, right? So we have to understand what partial pressure of oxygen speaks to before we can interpret the partial pressure of oxygen, right? So could anybody else give rise to what partial pressure of oxygen speaks to? What does it speak to?
The basically the amount of oxygen available.
All right. The since oxygen is technically gaseous, right? It's usually gaseous. Its standard state is gaseous, right? When we're talking about the presence of oxygen within a specific place, we're going to be using its concentration. In the case that its standard state is gaseous, then we use pressures instead of concentrations, right? So the partial pressure of oxygen kind of speaks to the amount of oxygen within a specific area. That's why when we look at here, when we have high partial pressures of oxygen, this is usually an area within the lungs, right? And down here is usually within the tissues, right? So to understand the implications, the physiological implications of what's happening within this area here, right? So we hinted to it before. We are speaking about efficient oxygen unloading, all right? So we're looking at that. So how is that important for mammals specifically? The importance of that part of the curve for mammals, right? So what we're saying now is that, as we were stating before, right, any type of drop in the partial pressure of oxygen would lead to a significant, right, in well, a significant release of oxygen, and any type of increase of partial pressure of oxygen within that region, right, will lead to a significant uptake of oxygen, right? So we're looking at this, the how the saturation will change within this region. So the efficient oxygen unloading is important here. So any small change in the partial pressure of oxygen will result in a significant change in the hemoglobin saturation. This allows for efficient unloading of oxygen to where it is needed, right? So that's important in specific, in mammals, right? Because what we are looking at is that when we reach tissues that have been metabolizing, tissues that have been respiring, right? Because the partial pressure of oxygen would be a bit lower, right? Hemoglobin's affinity would drop drastically and allow the release of that oxygen, right, in order to replenish the cell supply of oxygen, especially as the final electron acceptor in complex 4 of the electron transport chain. That's important there, right? So it's especially important for effective oxygen unloading, right? And it will lend in the tissue. It will lend into rather tissue oxygenation, right? So periods of high metabolic activity will actually shift the curve, and that will release oxygen in order to oxygenate the tissues, right, efficiently. And it also is responsive to any type of physiological change, right? So we're looking at the Bohr effect as well. That region is highly sensitive to the partial pressure of oxygen. So any changes within the partial pressure of oxygen, right, the Bohr effect will actually move the oxygen dissociation curve specifically to counteract these changes, right? I'm just generally explaining generally how that works. So within this region, right, it speaks specifically to the ease of oxygen unloading and efficient tissue oxygenation, right? It's really around 3 marks, right? So just look at the importance of that curve, right? Between the partial pressure. So when there's a slight drop in the partial pressure, it will affect the affinity of hemoglobin for oxygen, right? So what we're looking at is the efficient unloading of oxygen and tissue oxygenation, right, within those regions. Okay, and once it's increasing just a little bit, hemoglobin will actually bind more stronger to oxygen. All right, so we're generally just looking at those things for that question. For this question, no, it's generally speaking about the same thing, but we're looking at a shift. So figure 6 shows the oxygen dissociation curve for the adult hemoglobin at low carbon dioxide concentrations in the lung capillaries of a mammal, and the percentage saturation of hemoglobin with oxygen at the various partial pressures in an active muscle where the concentration of carbon dioxide is high. All right. So we're looking at the unbroken line is where we're looking at the lung capillaries, and the broken line with the dots is what we're looking at with the muscles, right? So the loading of oxygen onto hemoglobin takes place where oxygen concentrations are high, right? Look at the question, just giving you information, right? The unloading of oxygen takes place where carbon dioxide is produced and oxygen concentrations are low, right? So this is basically saying, hey, here's information here, free information in the middle of the exam. All right. So that is some information that could possibly be used. So what we're looking at here, right? The unbroken curve here is generally what we've been looking at above, right? Generally just the oxygen concentration, partial pressure of oxygen in relation to saturation of hemoglobin within lung capillaries, right? And we're having now a shift to the right, and this is how it looks within the muscle capillary, the muscle cells, right? So the muscle cells now, well, we're gonna be looking at that. Okay. So, what does the Bohr effect do? We're going to be speaking about the Bohr effect and what's happening there, right? It is also important to note here that the pH in the lung capillaries are alkaline, right? Well, both of them are alkaline. This is 7.4, and this one is a 7.2. And could anybody just reinstate what is the Bohr effect? Tell me what the Bohr effect is. The Bohr effect speaks to generally two to three conditions and what the effects of those conditions are. Explain to me the Bohr effect. Anybody? Before we even move on to the question here, explain to me the Bohr effect.
Um, can I go ahead, sir?
Go ahead.
All right. So the Bohr effect essentially is describe the phenomenon where hemoglobin's affinity for oxygen changes depending on the condition in which the hemoglobin is. For example, its affinity for oxygen is inversely related to its the carbon dioxide concentration of the blood, and hence, you know, if the carbon dioxide concentration increase, the affinity of hemoglobin for oxygen decreases. Similarly with the blood pH and temperature, and yeah, I think that's it. Blood temperature, and yeah.
Okay. Really, that's interesting. When we're having a right shift, what do we have increasing? CO2 concentrations increase, right? Work with me, guys. What's happening? CO2 concentration increases, yes?
Yes, sir.
All right. The pH, what would it do? Increase or decrease?
It would decrease
What would happen to temperature?
it would increase
Why?
because the metabolic reactions create heat as a byproduct.
Okay. And all of them are flipped if we shift to the left, right? Okay. Okay. Lovely. The Bohr effect explained. Let's move on with our lives. So label the relevant Are you serious? Okay. Label the relevant curve in figure 6 where you expect the loading of oxygen to occur in the lung capillaries and the unloading of oxygen to occur in the active muscle cells. So where do you expect the loading of oxygen to occur within the lung? Right, so within the lung capillaries. The loading of oxygen. Where would oxygen come on? Give me the numbers that you would use on the partial pressure. Well, on the x-axis, rather. Where would loading take place?
Sir, about um in the lungs,
Yes.
about 2 kilopascal.
Give me a range. 2 to around 5 kilopascals, right?
Yes, sir.
So you're saying that if we're looking at the capillaries within the lungs, the saturation of hemoglobin, well, saturation of hemoglobin with oxygen would increase. So where it increases, this general range, right, that we're talking about. I guess I have to erase everything on the screen. All right, to make it easier to see. I we just discussed all of that already, so that's fine. All right, let's make this easier to see. Okay, so when we're talking about the lung capillaries, right, we're saying that here is generally we're gonna have the loading. Let's put it here, right? So here we see that the saturation of oxygen will be increasing, right? So this is the lung capillary, so the saturation will continue to increase until it plateaus. Now within the active muscle cells, where with unloading of oxygen occur? Where would oxygen start to unload? Let me just read the question once more. Where would the unloading of oxygen occur? Oh, it says and the unloading of oxygen, therefore it's one area. Technically, the unloading of oxygen would occur within the same region that you stipulated. All right, because technically one curve is going this way and one curve is going this way. Because in one we're looking at oxygen being taken up in the lungs, and in the other curve we're looking at oxygen being given off, right? So we're looking at that. Does that make sense?
Yes, sir.
Okay, it makes sense to you. Does it make sense to everybody else? Hopefully that makes sense. I mean, we're just trying to understand the concepts because obviously the question will not repeat itself, right? But we're just trying to understand the concepts. All right. State the name given to the whoa. Moving on. State one significance of the increasing effect of carbon dioxide concentrations on the efficient delivery of oxygen to an active muscle while someone is swimming. So state one significance of the increasing effect of carbon dioxide concentration on the efficiency of oxygen delivery in active muscle cells. Right? The rest of it is just fluff. We don't care. Right? What is happening here? What's the significant effect? By the way, it's the Bohr effect for anybody who never realized why I just go across the question. This is not even a real question. The Bohr effect. Let me just put this the to be sure. Okay. But down here, state one significance of the increasing effect of carbon dioxide concentrations on the efficient delivery of oxygen in active muscle cells. Wasn't the link for this sent out already? Persons don't have the link. Okay, go ahead. Anybody? All right. So I'm not sure if anybody has a response here. Okay, let me see. I see a response in the chat. All right. So the oxygen will dissociate from hemoglobin to supply to the muscles that need it. I mean, yeah. Okay. Okay. So state one significance of the increasing effect of carbon dioxide. So as carbon dioxide concentration increases, we have the creation of carbonic acid, which will decrease the pH. Right? And the Bohr effect states that it's gonna counteract that. It's gonna counteract that change. The the affinity of hemoglobin will change, right, as the carbon dioxide concentration increases, right? So we're looking at that the affinity of hemoglobin for oxygen will change, right? So it will decrease the affinity its affinity for oxygen, releasing oxygen towards the active muscle cells that are respiring, right? So we're looking at the significance of that. Okay, just the Bohr effect. That's all you need to know for that. An investigation to examine the effect of exercise on blood pressure and heart rate is being planned. Two groups, each consisting of 5 16-year-old females, will be used. In the first stage of the investigation, Group A will train for 4 weeks swimming every day for 30 minutes, and Group B will train for 4 weeks cycling every day for 30 minutes. You are given the following apparatus to investigate the effect of exercise on the blood pressure and pulse rate at the end of the first stage. Alright. So we have the electronic pulse and blood pressure detector, we have a stopwatch, and we have a stepping stool. Wow. Outline a suitable procedure for investigating the effect of exercise on blood pressure and pulse rate for the two groups. What would be a suitable procedure? 5 marks, at least 5 steps. So let's go. What's a suitable procedure for this? How would we test, right, the effect of exercise on blood pressure and the pulse rate? We have a stepping stool that is 45 centimeters high. Go ahead.
Excuse me, sir. I attempted this earlier, but I'm not sure if my procedure is right. Could you check it? I can send it in the chat.
Okay, you can send it in the chat and we'll have a look at it.
Okay, sir. Thank you, sir.
I'll put it on the screen and we'll scrutinize it.
All right, sir.
Okay, so when you send it, let me know. We'll have a look at this. What a beautiful procedure. 3 steps alone, or are you sending more?
Oh, sir, there's a second part.
Okay, lovely. Let me paste these on the screen and we'll look at it and make any amendments to it. Alright, let's see what the second part is. 4, 5, 6. Okay, that should be fine. I mean, the 6th step is almost always repeating it or recording all the results obtained. Okay, so here we have a beautiful procedure. Let's read through the procedure as if we were a CAPE teacher looking at somebody's labs. Okay, so now step 1, at the start of the 4 weeks, before the exercise starts, let each student sit on the stool and take their heart rate and blood pressure using the electronic pulse and the blood pressure detector. Do this for 1 minute. Alright, why do we do it for 1 minute?
Sir, so that we can have an idea of how of the person's heart rate and blood pressure in a controlled time.
Define heart rate.
Sir, sir, the number of beats per minute that the heart makes.
Lovely, that's how we do it for 1 minute. Ooh. Look at you putting in the correct information. Alright, lovely. So that sounds pretty good. Do it for 1 minute so we can actually have an accurate heart rate. So if you don't do it for 1 minute, you're not gonna really have an accurate. Well, technically you can just expedite it using some mathematical gymnastics. But it's better to do it for 1 minute, right? You can generally do it for like 5 minutes or so to make sure that you're consistent. Average the values. That will give a better and more consistent, value. Alright? Because if you did 1 minute and there was an error, the systematic and parallax error will just stay there. Alright? So it's better you do it for like 5 minutes or so, and then you can boil it down through calculation of the heart rate and etc. If you get what I'm saying. Does that make more sense?
Yes, sir.
Okay. Step 2, make each student exercise for 30 minutes and time the period using the stopwatch. Alright, so we have that. Then at the end of the period, take their blood pressure and the heart rate for 1 minute immediately. So let's do it for 5 minutes. Let's keep through the procedure immediately after, right? And let them rest for 15 minutes, right? Time this with the stopwatch and then let each person sit on the stool. Okay, so we're seeing it done. So hopefully everybody can understand the significance of all these steps. These steps are pretty good.
Question, sir.
Go ahead.
Do we have to always write in present tense?
Um. Okay. Do we have to write in present tense? What we're doing is planning a procedure, so it hasn't been done yet, so it's gonna be in present tense. Yeah. No, the thing is that
I'm supposed to write it in past tense.
past tense. Supposed to be in past tense.
I don't know. I'm asking.
Okay, so the thing is, that caught me off guard, because usually you'll just write it in present tense. It's like a PD. With your PD procedure, do you write it in past tense?
No.
Okay, so I thought that you just outline a suitable procedure. So,
Other that,
go
we
ahead.
just do unit 1 bio, but the way they asked the question, it seemed like you did the experiment already. So I just wrote it in past tense.
Oh, okay. Okay, that makes sense. That makes sense. Cause it seemed like it was done already based on the experiment. Okay, that shouldn't be too bad. But it shouldn't be too bad though. right? Just writing notes in present tense shouldn't be too bad though. We're saying here that would repeat all steps 1 to 4 and then. Oh, so we're gonna repeat steps 1 to 4 for each group of students, right? And then do it after the 4 week training period. 4 week needs a hyphen. That's what it's called. All right. So we have that there. I think this is okay. Because what we're doing from step 1 to step 4, we're really measuring the heart rate before they have done some work, after they have done some work and started respiring, right? Like immediately, well, let me just rephrase that. We're measuring it before they have done any exercise, right? We're measuring it just right after they have done some exercise, and then 15 minutes after they have done the exercise, right? So we're looking at it at both different times, right? So looking at it before, during, and after. So we're having accurate measurements of the effect of exercise on the body, right? And that is before they have done any of the training regimens stipulated. And in step 6, we'll say that you will do this again after the end of the training period, right? So you will see the general differences, right, between them. I think this procedure is fine. This procedure is okay. Think it is fairly okay. All right, because you'll be able to get out everything that you need. You're using all the apparatus. All right, everything should be fine here. Because usually for this lab, I think this lab in module 3, I think almost everybody for unit, as long as you do unit 2 biology, I've never heard somebody not doing this lab already. So you guys would have done this lab. So there are different ways of doing it. Generally, do it before, during, and after, right, to measure the effect of exercise on the blood pressure and heart rate. Okay. Hormones are essential for control and coordination in animals. They are responsible for homeostatic systems, right? Involved in the maintenance of blood glucose levels. List 4 general principles of hormonal action in animals. What are the principles of hormonal action?
I think first we would talk about how it's produced. It's produced by ductless glands. All hormones are produced by ductless glands called endocrine glands.
Okay, producing endocrine, so they produce, right, in endocrine glands. Okay, so they're ductless, I guess. All right. Anything else? Any other principle? Okay, we're starting there. What else do we look at? Yeah, so 4 things. Let's go. Nobody else? Go ahead.
Sir, is it that they always have a target organ?
I mean, they would always have a target organ, right? Um, for the hormonal response. Yeah. So they'll always have a target organ. Do I want to put that right there? Um, let's see if we can rephrase it a little bit better. But let's look at this, right? The hormones produced um has an effector, I guess, right? Or what we call a target system. I mean, it doesn't have to be an organ per se, but let's say a target area, right? So it's a target area, a target organ, an effector. The term effector will cover all things, right? Everything. Go ahead.
So I was going to say that hormones are transported in the blood. So that's one of the principles of how its action is.
All
And
right.
I don't know the another one.
Why did primarily, right? Okay. Anything else? And I think the target effector specifically, I think that will lead into the hormonal specificity. right? And there's some amount of feedback regulation, or what we call mechanism. There are feedback mechanisms, regulations mechanisms, whatever you want to call them. Usually mechanisms, right? So they're produced by endocrine glands, right? These hormones are produced by endocrine glands. These hormones move via the humors of the body, primarily the blood, right? And they are going to be specific. They have some amount of hormonal specificity to a specific effector, right? And they are regulated via feedback mechanisms. Lovely, right? State two general principles regarding the homeostatic how homeostatic system works, right? So two principles regarding how a homeostatic system works. This question is kind of weird because didn't you just ask for all principle like four principles of hormonal action and looking for two of how homeostatic system works? Okay. Anything? Go ahead.
So I was thinking that for homeostatic system we could mention that it we could mention incorporate something regarding set points. So say something like the system works to counteract a change to the set point. So basically negative feedback. Yeah, that.
All right. So have that there. The feedback mechanisms there establish and maintain suitable um that should be ranges, right, close to set points, right. You could speak about temporal dynamics or what we call the time effect. I don't know, that would be a general principle though, the fact that hormonal responses can actually last over a huge amount of times, right. So hormones released an hour ago can affect you for like the next 5 hours, right? So they have huge, well, they have a long lasting effects. But that would be like a general principle for the question above. Um, anything? So generally the principles underlining are that homeostatic systems are through hormonal action and feedback mechanisms. Right, so this question kind of weird. It's kind of weird, but like, it's generally the feedback mechanisms and its integration with physiological systems that we talk about. Right, so because the homeostasis involves the integration of multiple systems. Right, I don't know if we could speak about that of multiple physiological systems rather systems, right? So we're looking at the fact that it's usually homeostasis usually works by an amalgamation of multiple different systems, right? You have different detectors and receptors all over the body that help to really counteract any type of change to keep the body in homeostasis. Go ahead.
We could, as a principle, I was thinking that the fact that the system in the body has to be constant, so like conditions in the body have to remain constant, because how we define homeostasis as, maintenance of a constant internal environment. So I would use, if me couldn't find nothing else in the exam, that as one of the principles.
Okay. Somebody raise their hand. I think Riri, person named.
Sir,
Go
I
ahead.
was gonna say negative feedback and positive feedback, but what he said make more sense.
I mean, yeah, okay. So negative feedback and positive feedback would be the feedback mechanisms to maintain a constant environment. Well, internal environment. The word internal is key. Alright. I think my brain just flashed back to like fifth form a while ago for some reason. Okay. Alright, let's go. So explain the rules of receptors, regulators, and oh my gosh. This is more like an. This question makes me feel like I don't want to do the paper anymore. Look, all 9 marks. It's not like you can't answer the question because the question is fairly easy, but 9 marks. Like you just feel like we just drop the pen and decide to say nah, go do it anymore. But okay, receptors, regulators, and effectors in the homeostatic system. Sure, explain those rules. You must, right? In your response, you must use an example which references the glucose concentration in the blood for each role. Okay, so basically, how does the pancreas, right, really help in the regulating the glucose concentration? What we're talking about is receptors. What are receptors? What are receptors? Go ahead.
Sir, systems or organs that detect and transport changes in an internal environment.
Okay. Anybody else? Because that's a lovely answer. Anyone else? Okay. All right. So looking at that, so the receptors, right? We know that we're gonna have specific cells, right? So they're gonna be glucose receptors, right? And they and what we're gonna be looking at. What are the main receptors within the glucose regulation? What detects it?
Sir, liver cells?
Go ahead.
Sir, liver cells?
That's interesting.
Yep.
Do they?
Sir, is it the alpha and beta cells?
Yeah, but they're not in the liver for the most part. So the hypo Okay, but the hypothalamus, right, would basically detect, right? It can detect glucose any changes in glucose concentration. Yeah, well, let me just explain what I'm doing before I do it. I'm tying the receptors and everything to the role, right, within the glucose concentration main regulation, right? Because that's what the question asks for. So the receptors will know. I'm just gonna discuss the question, right? So the receptors will pick up any type of change in internal stimuli, right? That goes past above or below a set point, right? Or that region there. Now, the main receptors, right? We have the hypothalamus that will play a role, right, within the detection of different blood levels, well, blood glucose levels, right? But the beta cells also can detect the difference within the blood levels, right? So looking at the receptors, they will receive these stimuli. What would be the regulators? What are regulators?
I think these are the well, I wouldn't say component, the part of the homeostatic system which coordinates response and compare the change to the set point to see what kind of response will happen.
All right. So now we're gonna be looking at those regulators, right? So the regulators will basically take in the information from the receptors, right? And now create an impulse, right? That will see how it will change the stimuli to see if we can return it to the original set point, right? So the regulators will actually, if it is a nerve, it will shoot an action potential. If it is a ductless gland, like an endocrine gland, it will release a hormone, right? And then those things will go to effectors, which will now carry out the entire thing, right? But what would be the regulators in the case of the glucose concentration regulation? What would be the effectors? Go ahead.
Excuse me, sir. Did you say effectors?
Not effectors, my bad. Regulators, my bad. Regulators.
Oh, okay.
Mm-hmm.
Sir, it would be the pancreas.
Be more specific. What's inside the pancreas? Where inside the pancreas do we find these areas? Start with an I.
Sir, I'm not too sure.
The islets of Langerhans. Right? So remember the islets? The islets of Langerhans, right? And within the islets of Langerhans, we have the different types of cells. What are the two main cells we look at? The. Am I losing anybody? Hopefully
alpha
not.
and beta.
The alpha and beta cells. So it's the alpha and beta cells located within the islets of Langerhans, within the pancreas. Those are regulators. What would the beta cell produce?
Insulin,
So what do they detect?
high glucose concentrations,
Elevated glucose levels. Lovely, right? And what would the alpha cells detect
low glucose concentrations,
and produce?
glucagon,
Lovely, right? Those are the regulators. So the receptors will help to really detect these things. The thing is, the receptor in this case would generally be, if we're talking about something outside the pancreas, we're talking about the hypothalamus for the most part, right? But within the pancreas itself, these cells can actually detect as well. They can recept as well, right? And they also regulate by producing their different hormones. So insulin produced by the beta cells will help to decrease elevated blood glucose levels, and glucagon secreted by the alpha cells will help to increase any type of lowered blood glucose level, right? And what are the effectors? What will now take these hormones and do change with it? What are the effectors?
the blood,
No, not necessarily. Hormones are transported to the blood, but where do these hormones go? What are the effectors which will now do the change? Go ahead.
It would be muscle cells or liver cells. Well, morely muscle cells.
Okay. I think Suki was um unmuting at all. Don't know if you want to say something.
So I was going to answer. So
Okay.
it's okay.
So let's say okay. So the liver, the muscle cells, all these respiring cells, adipose cells, those cells are going to be the effectors. Adipose cells will allow for gluconeogenesis to occur, glycolysis, and these different types of things. And well glycogenesis and gluconeogenesis and those type of things, right? Usually gluconeogenesis for the most part will occur, right? And the lysis of glycogen, the breakdown of glycogen will occur within muscle cells and stuff like that, right? And within the liver cells, we're going to have an entire cycle that occurs there. The liver is a huge organ that helps with the breakdown of different materials, right? So the liver is basically like the trash compactor of the body. It breaks down everything, right? Your cell is dead, it breaks it down. Your blood cell is dead, it breaks it down. Glucose is there, well, not glucose, but glycogen is there, it's gonna break it down, right? So it's going. Those are the effectors: the muscle cells and primarily the liver, right? So, well, let me switch that, right? Primarily the muscle cells and the liver. Let's say that, right? Those are the effectors, and we must basically speak about these things in cohesive paragraphs. So, once we understand the concept of receptor, regulator, and effector, all we have to do is tie it to a response, right? So, if they brought this now and asked for the osmoregulatory response, right, what would be the receptor?
the kidney.
The receptor?
Wait, no.
Sorry?
Repeat?
Yes, sir.
No, I didn't hear what you said. Repeat.
But I'm asking you to repeat.
Oh, what if we were talking about osmoregulation? What would be the receptor? Because this came last year. Suppose osmoregulation come next this year, or suppose they didn't bring hormones at all. I don't know, but I don't know. But what would it be for the osmoregulation? It's gonna be the hypothalamus again. So know that, and those will not change. Once you know the hypothalamus is the receptor, it's generally a universal receptor. It's gonna be the hypothalamus again. What are going to be our regulators? The pituitary gland, the posterior pituitary gland will be the regulator that will produce the ADH. Mm-hmm. And then our effectors would be what? The cells where?
The cells, the epithelial cells of the collecting duct
Lovely.
and distal convoluted tube.
And the distal convoluted tubule. Lovely. So we must know them. Alright? So if anything like that comes, you should be able to tackle that. And we're brought towards module 3.